Good morning, everyone. Welcome to Pediatric Grand Rounds. It's a pleasure to welcome you to Discovery Day today. It's an occasion that allows us to celebrate the pursuit of knowledge and innovation in child health research. So a day to sort of celebrate what we do and the difference makers for us in our department, in our hospital. Today, we have the distinct honor of hosting a visionary leader, a wonderful pediatrician, a forceful advocate for children's health, and my friend and colleague, Dr. Phyllis Dennery. I just want to tell you a few words about Dr. Dennery. She is the Sylvia K. Hassenfeld Professor and Chair of Pediatrics at the Brown University Warren Alpert Medical School, the Pediatrician in Chief at Rhode Island Hospital, and the Medical Director at Hasbro Children's Hospital. She also is Professor of Molecular Biology, Cell Biology, and Biochemistry at Brown, and likely attends more faculty meetings than anyone I know. A consummate physician scientist, Dr. Dennery studies mechanisms of neonatal hyperoxic lung injury with a particular emphasis on the role of oxidative damage in altering lung metabolism and architecture. Her clinical interests focus on the long-term consequences of prematurity. She enjoys many collaborations which collectively bridge important gaps in knowledge and foster a broad commitment to child health. Dr. Dennery's scholarly impact is substantial. She has authored or co-authored more than 130 original scientific articles, reviews, and book chapters, and she has been continuously funded by the NIH. In acknowledgement of her many contributions, Dr. Dennery has received several awards for her research and her mentoring, and she is a member of the Association of American Physicians and the National Academy of Medicine. Dr. Dennery earned her BS degree from McGill University and her MD degree from Howard University. She completed a residency in neonatology at Case Western Reserve University and served as the director of the Division of Neonatology at the Children's Hospital of Philadelphia until she was recruited to lead the Department of Pediatrics at Brown about 10 years ago, which is about a year before I became chair here. How time flies, Phyllis. One of the most remarkable aspects of Dr. Dennery's career is her commitment to the field of pediatrics and the training of pediatric physician scientists. She chairs the steering committees of several multi-center studies and is a past president of the Society for Pediatric Research. She is a member of many boards and committees focused on pediatric workforce development, including the external advisory committees for both our NIH K-12 and T32 training programs, for which we are mighty grateful. She is now co-chair of a National Academies Committee on strategies to enhance pediatric health research funded by the NIH, and we wish her great good luck with that work. This morning, Dr. Dennery is going to discuss her research and her career as a physician scientist. Today at 12:30, I have the pleasure of interviewing Dr. Dennery, and she's going to interview me a little bit too, and we're going to talk about careers and the future of physician scientists. That's what we're going to do later today. But first, we're going to hear her Discovery Day lecture, which is entitled A Physician Scientist Odyssey. Is there a safe path to success? Please join me in welcoming Dr. Phyllis Dennery to UPMC Children's Hospital.
Good morning. I felt like I was going through a trap door or something back there. But thank you so much for having me here. And I give this talk in various formats, and I decided I'll theme it on Greek mythology. So forgive me if this is a little corny. But here we have Ulysses fighting off the enemies, and this is what we're going to have to do a little more. Oh, yes, thank you. This is what we'll have to do in science and in trying to continue to advocate for science, as you well are aware. So typically the path to success for physician scientists aligns like this. You do research, publications, grants, talks, promotion, and tenure, and then you should be fine, right? But there's a lot of things along the way, and as you ascend into the light, sometimes that can be challenging. Right now, for pediatrics, we have a problem. We see less and less folks interested in a career in pediatrics. Even though this year's match was better than last year's, as you are aware, it's still a declining trend overall for interest in pediatrics, which seemed to peak in 2015, but then is gradually going down. And this year, we were thrilled that the fill rate for pediatric residencies went back up, but not even quite to the rate that we were previously. So the problem is that we have does this show as a pointer? No, I don't think so. That this, see if I can find a pointer. Maybe that could be helpful because I tend to point. Okay, here. Yeah, that's visible now. So we are still at lower numbers than we should be. And last year was very terrifying because we dropped significantly. So that doesn't just apply to categorical pediatrics. It's for subspecialties in pediatrics as well. If you look at this, you can see that there is, why is this not going here? You can see that most of the subspecialties of pediatrics have less than one applicant per position. So that's really challenging for fellowship programs. The only ones that seem to do okay are hospitalist medicine and cardiology. And on the cusp are critical care, neonatology, and emergency medicine, although this year there's a lot of interest in emergency medicine. And everyone else is less than one applicant per position. And unfortunately for infectious disease, Terry, it's pretty bad. So I think we have to revitalize the excitement around these subspecialties, but we're not there yet. So as I said, many subspecialties find themselves at a loss, and physician vacancies in pediatrics are enormous. We try very hard to recruit neurologists, behavioral pediatricians, genetics, etc. Pulmonology for us is a real challenge. And you can see that folks are deciding otherwise in their careers. To compound this problem, we have a challenge, and folks who are doing subspecialty usually are interested in conducting research. And this is what we see. There's less funding, so there's more applicants, but less funding for research, or just a modicum of increase. And now I'm not sure what to tell you what's going to happen next. But this is really a problem. And if you look at who gets funded, well, now some of these are no longer in existence, but there is a lower rate of funding for things that we care about: child health, minority health, etc. Those are much less funded than the rest of the others. And here, right here, which is fascinating to me, is the Institute for Eye Disease. But that's where the funding goes. So we have a ways to go. So I'm not trying to depress you today, but there's a few more things we have to talk about, which is that there is a challenge, poor Sisyphus here trying to push that rock up the mountain. This is what we're facing. We have science under siege. We have threats to cuts, and people are fighting as best they can to push these things off. But what the resultant thing is, for me, on a daily basis, I fear that we will not have funding, and that folks will get discouraged about careers in pediatrics, in subspecialties, and it'll be very tough to continue to make strides forward. And to make matters worse, we have this banning of words, right? And so I thought this was our Trojan horse, that we could find substitutes for these words. Most of these words are triggers that can, eliminate your application. We've had that happen. But now AI has found ways to overcome these words. It sounds silly, but I think it's okay. Don't tell everybody. But there is definitely a way to say things like biased. Instead of saying bias, they don't like that word. So you can say prejudice. Somehow that doesn't trigger. And community diversity, you can say variety in groups or something. So it goes on and on and on. You have equal opportunity, fair access, or whatever. It's all the same, but it seems like we have to play these crazy games. And unfortunately, some of the, and these are, you can find this online. It's actually quite interesting. Some things, it's really hard to change saying certain things, right? So we have to find riddles and ways to say it. Race. How do you say race other than race or ethnic group? Sexual preferences. That gets flagged. Women. Wow. So all these things become a trigger and may flag your application and then cause it to be removed. So that leads to people feeling somewhat discouraged. And in recent years, many people who are coming along and wanting to be trained in research go through the process of getting funding. And one of the funding mechanisms, of course, is a K award. And it looks like, in fact, women are doing less well than they were in terms of getting funding through the K mechanisms. And there is also discrepancies in terms of Black and Hispanic applicants getting the funding. And but K awardees are more likely to get an R01 or an R01 equivalent over time. So it's, and the support here for chairs, this is a real problem. They only give you $75,000 to $100,000 for 75% effort. I think that's a little difficult in terms of matching the amount that we need to get to pay someone and the amount that they give you. So, but there's still a benefit to getting this so that you can move on in your career. Unfortunately, many of the folks say that it's not what they want to spend all their time doing, working, being crazy busy, not having work-life balance, those types of things. And they want more flexibility. They want to do different things. And we'll talk more about that later. But there's only so many hours in the day, as you know. And I think that we often wonder if we're, setting ourselves for a life of misery and pain. But I will argue that, and well, I'll get to this first. There's, and people worry, am I going to find a job? Am I going to get paid enough? Am I going to be balanced in my life? And people might choose to leave this path, but I would argue that it's fun. Research and academics are fun. We don't have a daily routine that's rigid. It depends on your personality, but for me, the idea of every day having the same thing over and over again is difficult. You meet a lot of bright, wonderful people. You can think strategically. You can make influence at a greater level. You can mentor. That's one of the joys of my life, is mentoring, being able to talk to trainees. And sometimes you get to go to Pittsburgh or something. You get to travel. And here's Bacchus enjoying life and having fun. So there's so many things you can do as well. As we know, I'm just showing you many examples of ways in which people can get engaged, including AI and quality improvement. And you don't always have to be in the lab pipetting as I do, but you could do simulation research. This is CHOP's idea of the artificial womb with lambs and so forth. We'll see how that goes. But there's many things you can do to enhance the knowledge of the community. And how do you become successful in research? I don't have all the answers at all, but one of the things that are known to make a difference is if you build a network, if you have persistence, resilience, which we'll need a lot of, initiative, autonomy, and some balance in your life. And I'll remind you that a pessimist sees the difficulty in every opportunity, and an optimist sees the opportunity in every difficulty. So let's be optimistic and grateful that we have these experiences and this training and so forth. There was an article written by one of my trainees, Christina Alvira, who wrote that what does it take to be successful and what drives strategies for success? So time is a most important commodity. Work-life balance, mentorship. We'll talk a little bit about that. Lean in, aim high, speak up. Don't be afraid to speak up. This is actually very pertinent right now. Enlist leaders to achieve equality, be optimistic, tenacious, resilient. Tell your story. People like to hear how you overcame challenges and whether you have things you still worry about, because we are all humans and we want to be able to speak about those things. So this was an article recently about the impact of building community. This can apply to any community. This was around the community of practice, but you build a network, you expand, you exchange ideas, you just work further and further into elaborating your sphere of influence in the community and elsewhere so that you can become more successful. So I'll take a little route through how I first started. I'm a neonatologist, so I was passionate about neonatology. I was excited when I was a third-year medical student. I had the opportunity to witness the NICU, and there was this tiny baby. And in those days, people weren't as good about hand washing and all that stuff. So the attending put her finger into the isolette, and the little baby just grabbed her finger. And I went, wow, this is amazing. To be able to have that small of a person become a big human being with all the things that we do, and they start off so small and they succeed. So I said, I want to be a neonatologist, but I first had to be a pediatrician. And I did my training at Children's Hospital in DC and learned to love kids of all ages. But I still stayed focused on neonatology, and my interest was really around lung disease, seeing the lungs get damaged. In those days, that's the early 80s. There was a lot of damage from ventilators. We didn't quite know what to do. There wasn't surfactant yet. So these lungs would get more and more damaged. And now as we resuscitate smaller and smaller babies, that's even the case now with whatever we do. So I was leaving my fellowship training at Case Western, and I had done some work on oxidative stress in cells and culture, and I was going to go to Stanford University, and their interests were around bilirubin and kernicterus and the damage that kernicterus can do to the brain and how it impacts our children. And David Stevenson was my division director at the time, and Hank Freeman worked in his lab on different ways to deliver light therapy for hyperbilirubinemia. And as I was walking out the door at Case Western, John Enever, who used to be the division director, said, have you ever thought of bilirubin as an antioxidant? How do you bridge what they do at Stanford with what you're going to want to do, which has to do with the lung? Is there a way to bring those two things together? And I went to Stanford, where I was introduced to Tony McDonough, who's the funniest man you ever met, but unfortunately he succumbed to liver cancer a few years back. And he was the expert who had these mice. He had these gun rats. These rats were, the homozygotes were jaundiced, and they would succumb from kernicterus. However, the heterozygotes were not particularly jaundiced, but they had a higher level of jaundice than a normal wild type would. So we said, well, let's take a look and see if they have an affinity and an improved tolerance to hyperbilirubinemia and to oxidative stress. So I also met some wonderful people from the NIH, Rod Levine and Doug Spitz, who said, okay, why don't you look at this oxidative stress in the lung, look at the levels of bilirubin and try to see if you can understand whether that's a beneficial impact on the lung. And lo and behold, the animals that were slightly jaundiced had an improved capacity against oxidative stress. And this led to more work around the fact that bilirubin is really the byproduct of heme, which is degraded by this enzyme heme oxygenase, which is the rate limiting enzyme. And at the time, there was a lot of work coming out about heme oxygenase being a stress protein. So we did for many, many years work on heme oxygenase and developed a track record in that field. The last bit of time when I was at Children's Hospital of Philadelphia and transitioning to Brown, we looked at heme oxygenase as a DNA binding protein, which was quite novel, and were able to understand that heme oxygenase itself can trigger different genes to be regulated and dysregulated. So that was a long track of way where at Brown I was able to meet some folks in computational biology and mathematical modeling who helped figure these things out. Again, pointing to the collaborations. You meet people, you work together, you develop new ideas. However, at that time, when I got to Brown, I was really realizing that my overarching question had to do with neonatal lung injury, how to mitigate neonatal lung injury. That's where I started when I was looking at it from the standpoint of a fellowship. And at Brown, there's a lot of folks working on senescence. And so is it something that could help us understand what's happening to the lung of premature babies? As I said, these lungs get damaged and there's a lot of damage that happens. So I'll resume this story a little bit later, but as I was mentioning to you, a lot of things that we do have to do with our mentors, the people who help us get to some new thoughts, to work our ways, make connections for us. And they come in all sexes, sizes, colors, nationalities, because if you are looking for someone that looks just like you, for some of us, that's a difficult thing to find. But we have to realize that many people care about us. And there are people here, I don't need to mention 1,000 names. I've talked about some of them, but one person in particular, Maria Delavoria Papadopoulos, she was a neonatologist of neonatologists. She started the field years and years ago, and we crossed paths at Children's Hospital of Philadelphia years ago. And she was such a powerful woman who really was a pioneer in the 50s and 60s when women didn't have many places to express their greatness. So the only other person I'm going to point out is Condoleezza Rice. I don't know if some of you still remember, there's a lot of millennials and things in here, but for us, she used to be the chief of staff. What was she? A big deal in the Bush administration. And she was also the provost at Stanford before she started that route with Bush. And she was, I didn't share her views necessarily, but she was there as a provost and she'd invite people to come to have lunch with her at her house. And when I went there to meet with her, she said, you know, a lot of people like us, Black women, will have a much different course because we often we get singled out a little bit to do something a little earlier than we would like to. So for her, it was that she was a Russian scholar. She was interested in Russian literature. And at the time, she was helpful to the Bush administration because she knew something about Russian culture and they were trying to fight the Cold War or whatever it was. And so she moved away from her academic position to become an administrator. And that happens to many of us. We get pulled away from the things we're good at because we're told, well, you're good at this, so let's make you do other things. So beware at times, you know, take your time. She, you know, I'm not sure what she's doing these days, but I thought that was a good message that she gave on that day. The mentors, let's talk about mentors, because you need mentors. Everyone can be your mentor. They can give you time, resources, sometimes advice, networking. That's really one of the key features. They can introduce you to people. If they're good mentors, they'll do all that stuff. And what you need really is time to do your work, institutional resources, look to Terry for that, and strong community around you to help you. If you come in a place and you're the only one there doing this thing, you may not really be as successful, because how are you supposed to start something with no one around to help you? So that's good when you make career decisions. We did this at Brown. We tried to really help people the best we could to be successful in their careers in research. When I first came there wasn't a lot going on. And so I appointed someone vice chair for research and faculty development. And she and I worked on creating a mentorship program where mentors and mentees were paired in dyads. And we were able to help them understand what grants are available, what programs are local at Brown, created a few internal programs for, pilot funding, and had workshops, grant writing, manuscript writing, a whole lot of things that can help people understand how to make it forward. We hired a biostatistician, et cetera. So we gave them an opportunity to find ways to ask the right questions. And so when we did this, we saw a big difference in our total grant funding. There was very little funding in the early years. And as we made changes, we saw a dramatic difference in how much funding was taken over. And now we continue to increase. We just got funded right under the line before all these changes occurred for T32. And so we are making strides in getting people interested in research. We have a research day coming up soon, just like you're having. And the productivity is really dramatically different. There's a lot of collaborations. There's a lot of awards. There's a lot of grant funding that takes place. And people write about this because they feel more comfortable doing. And we're trying to move that to an earlier stage where residents also can learn how to enjoy research. I'm sure you have a research track here. We didn't. So now we're trying to create an atmosphere where people feel comfortable taking on. This is an example in Italy where they did this early training program for residents, and they saw a tremendous increase in interest in research careers after the program was instituted. 50% of all residents interested in careers in research. So one big challenge we have is UIM clinician scientists. So there's very few people who represent minorities in as clinician scientists. And why? Well, it's interesting that contrary to what's going on in the world, where the most a large number of children are born are minority children, and soon they will exceed the number of white children. So this says by 2018, but I think we're close right now. Only 5% of medical school faculty, and that's a number that it has not budged in decades, and 3% of professors are minorities. And if you look at folks getting grants, only 5% of R01 applications are from African American, Latino, Native American investigators. And just like in Greek mythology, it's very hard to find diverse characters in there, but I found one. And so it's challenging to see where that would lead us. And what's even more problematic is that folks are less likely, ethnic minorities, Blacks and others are less likely to stay with academics and to stay with funding and research rates. So they're less likely to get funded, less likely to stay with it. So this, I'm sorry, this is a confusing slide right here. But the bottom line was that if you were more likely to leave academics, that's what I'm trying to say, if you were Black, and the other one's less likely. So that's what's really interesting, is that we are not seeing folks interested in these careers because they don't see a path forward. And if you look at the numbers of folks who are getting funded or K applicants for Black, it's very low. And that's really in contrast to, for example, whites and Asians, less so, but whites, Black numbers are staying fairly low. So, so what do we do about this? Well, as you know, there was an executive order that says we have to end wasteful DEI programs, that was signed as one of the first acts of Donald Trump's presidency this time around. So why are we so opposed? I don't know. But bottom line is that we have to, within our groups, find ways around this, ways to continue to be strategic, to let folks know that we're interested in seeing them there, rebranding some of the programs, expanding the scope, and looking for ways in which to continue to mentor and support people who are of underrepresented backgrounds. So this slide says research mentorship. One great program was the PSDP program, which just got eliminated. And that had an opportunity to engage people and to bring residents to see what research was all about. So that's a real challenge. And we'll find a way around it. I think they're appealing the decision, but the year before, when the grant was renewed, the first application said, well, you know, we cannot, you don't have enough content about diversity. So the grant was changed to reflect that. And now it's like, well, you have too much, so we don't want to fund you. So that's a real problem. And at Brown, what we did was we increased the recruitment because we had a targeted approach to saying, let's build a curriculum, social justice curriculum, have a diversity visiting scholars program, racial caucusing, which is an interesting concept where people from different races get in rooms together just by their race, which is scary, you think. However, it's to find solutions, to help people think through what is going on and how can you increase support for all participants and not ask the participants themselves to find the solutions. And so a lot of different initiatives, education on microaggressions, et cetera. And as a consequence of that, we've saw a huge number of incoming class being underrepresented, faculty also, promotions, et cetera. So that's been a great pleasure to see that happen. So there is hope for the future, despite all that I've said to you that sounds very tragic. There's a lot of folks fighting back. So justice departments are pushing back and saying, you know, we have to be able to continue. Research is key to our success as a nation. And that's the worry that we have if we stop that. And there are ways to support, and I will advocate for everyone to advocate, you know, support graduate medical education, invest in clinician training, expand the eligibility for loan repayment. Those are the things you can advocate for with your legislators, and increase support for the pediatric workforce through Medicaid, which is under threat, but support Medicaid and keep Medicaid expansion as possible. So you have a role to play to advocate. The more you show up, they tell us pediatricians are never there. Surgeons are there all the time. How do you get more of us to go to the Hill and to say, hey, work with us? This is the importance of why we do the things we do. So back to how to get ahead in research. I told you these things. You have to be passionate. You have to be ambitious. You have to be driven. A lot of people think ambitious is a bad word. I disagree. Ambitious doesn't have to mean blind ambition where you break everybody in your path. It has to be that you are focused on what you're doing and that you're going to continue to make strides forward. And until you make a commitment, you will only see walls. So you have to have that fire in your belly, that ability to see something bigger beyond the challenges. And how do you help your research? How do you make things work in your favor? This is Cyclops being blinded, as you can say. Don't ignore the data, that what the data is saying. So if you see something, but someone else published something else, but you repeatedly see something in your data, that's what it is. You can't make a story. You can't bend a story to meet whatever someone else said. But you do reach for others for their opinions, because it's important to look around and see what people are doing around you. So I would really recommend that people seek out other people's opinions. And a lot of these mentorship programs are just that. You have folks that help you get focused. And this one, don't believe everything you read. So sometimes people say, oh, this is the way it is, and you're afraid to stand up for something else. But if you don't, you will continue falsehoods, for example. So, I think the most important thing I've learned is to be humble, to seek advice from others in order to succeed. Ask others to review your papers, ask them to review your grants, get feedback on your projects. That way you'll have a chance at making it. And if you don't take a chance, you won't make that happen, that success happen. So many irons in the fire, as we say. So I tell the brief story, I like to say that, of some of my research grant history. So when I was at Stanford as a junior faculty, we didn't have, well, did we have K awards? I think we might have, but we had what's called the R29, which was essentially an independent, for your first stab at an independent grant research. And when I asked everybody's opinion, did all these things, I sent in the grant and I got this ridiculous score, 0.6 percentile. That's ridiculously good. And I was so proud of myself. I thought, oh, this is great. They advertised it in Stanford's, whatever. They said, never seen, never seen. So I thought I was so good. And then next time around, I submitted another grant, my first R1, and it got triaged. Why? Probably because I didn't ask anybody for their opinion, because I thought I was so good. And then I went back to asking for opinions to make sure I knew what I was writing, give people a chance to review. And by the way, don't give it to them to review the day before it's due. Ask them to review it well in advance so that they can be thoughtful about their review. And then I did okay, but certainly not as good as this. And on. We had times where we were triaged. Sometimes we got a grant. Sometimes we were triaged, got a grant. This is another story I like to tell, is this grant. The grant was submitted. It was just an R21, a small grant. And they essentially criticized a lot of silly things about it. They said, well, the investigator and their collaborators are not in the same institution. What? Does that matter? No. So I started getting really upset with that. And I at the time, there was an article that came out in PNAS, a fabulous journal, that said that folks like myself were less likely to get funded by NIH because there was some unconscious bias around funding. And so I wrote to the program, I told the program officer, I said, you know what? I think this had a lot to do with bias, because I don't understand why they didn't accept the grant. The guy was losing his mind. He said, oh my goodness, no. Just send it back in, add a thing or two, and you'll be fine. So it went from triage to the first percentile. So that tells you that sometimes you advocate for yourself. Not every time. I'm not saying every time you get a bad score, you go complain. But in this case, I mean, although it's tempting, in this case, you do, if you see something that's wrong, you might want to talk about it. So there were the ups and downs and ups and downs. On we go. But you're not going to succeed the first time always. Maybe some do, I don't know, maybe Terry, but others may not. And we have to be able to accept that. So another thing I like to do, and that's very important as you go in your career, is to pay it forward. You know, I was blessed by having a fantastic PI at Stanford, Dr. Stevenson. As I mentioned, David Stevenson was someone who promoted my career, gave me opportunities along the way. Sometimes it was a lot, but gave me chances at leadership, at engagement with others, etc. So I made it a point to always bring in trainees who then went on to do amazing things, being associate professors in Germany, being a professor here in this country, on and on, postdocs at Vanderbilt, on and on. So I was so excited to see that you can really help people build their careers and do things that are really very exceptional for them. So always look to those students who come to you and need support. And for some of us who are slightly older along the way, sometimes you reinvent yourself. So I mentioned, as I talked about transitioning to Brown, that they were interested in senescence and aging. And how am I, a neonatologist, going to get into that mix? Sometimes serendipity really makes you have an incredible discovery or redirection. And that's what happened in this case. So what we noticed is that oxygen, when it injures the lung, can lead to some cells that are senescent and aging. So that might change their lungs' function. So what we did was I hired somebody who was one of these experts in understanding senescence in my laboratory, and she looked at the impact of oxygen on cells in the lung, and lo and behold, there were marks of senescence throughout the lung. So that was an interesting observation that we pursued, because we thought, well, what if we used anti-aging therapies to help protect the lung against injury and maybe see some improvement? So how were we going to know which cells in the lung were more targeted for senescence? And what we did was we did what's called single cell transcriptomics. Maybe many of you know about this, and looked at the different cells in the lung and saw that these cells had different functions, of course. And when hyperoxia hit the lung, it had reduced progenitor cells, reduced transitions from type to type 1 cells, which are the reparative cells, et cetera. So that was the first step. And we decided to change this a little bit by, and I'm not showing you here, by taking the cells themselves and looking specifically for the senescent cells, and then allowing us to discover what these cells did. Interestingly, when we did this, we found that most of the cells, a lot of the cells that were senescent, were immune cells in the lung, macrophages in particular. And so with the help of folks like Jocelyn here, who helped us do this dissection of cells in the lung, we were able to find that only 11% of type 2 cells got senescent, but a vast majority of the cells were macrophages and some dividing cells. So what does that do for us? Well, we had to be able to then understand what these macrophages were doing. And so we further analyze the different clusters of macrophages to understand which ones were doing what. And some were protective, some were proliferative, and some were inflammatory. So then more dissection into this, we understood that these senescent cells can express what's called the SASP, the senescence-associated secretory proteins. So SASP is a collection of molecules that can promote cell immunity, wound healing, do some good things, but also do some bad things: aging, accelerated wound injury, fibrosis. So what are these cells doing? And so what we understood is, if we expose the cells to hyperoxia, took away specifically the senescent cells, we found that they expressed a lot of this SASP, Because if you think of the number of cells that are senescent in the lung, it's very small proportion. However, they're able to propagate their damage by producing this SASP. We did further. We found out what's in that SASP to try to understand how we could create therapeutics in order to then block that adverse event, for example. And we see a lot of different proteins, but one pathway that was accentuated was the SRC kinase pathway. So we're now going into trying to understand how we can develop therapies using the inhibition of the SRC pathway shown here to block that senescence. Because when we test this SASP on lungs, so we expose cells that are macrophages to high oxygen, separate out the senescent cells with this compound, and then we give it to the lungs of mice, which are not exposed to anything, and then we euthanize these mice later. And so what we did observe is that if you gave the SASP that had come from senescent cells to the lungs of these mice, they had worsened lung architecture. So decreased radial alveolar counts, increased mean linear intercept, and there are fewer blood vessels. So that suggests that whatever's in there can do a lot more than just damage the lung in very specific cells. So that was an interesting observation. And one last thing I'll bring up briefly is that we also found that there's a natural benefit to senescence. So if you look at the lung over the course of time, there's less senescence in the normal lung, not exposed to anything. But there is some peak in the first few days. What's that doing? Well, it turns out that in many tissues, senescent cells are there to remold, to change. So if you think of the lung, it's like a blob at first, and then you have to form units. And the senescent cells break up the things that shouldn't be there as the lung matures to allow for more diffusion of air and oxygen. So, so that's what we're focusing on. We are trying to understand how, if you disrupt this normal senescence by doing things like hyperoxia, what exactly happens and how can you mitigate it? It seems like it's based on P21. So developmental senescence causes all these problems if you interrupt it. So if you take the normal lung, you block senescence, in the wrong time in development, you can prevent normal lung alveolarization. So it's going to be important for therapies not only to worry about using an agent to kill senescence, but the timing of it. So if you interrupt the normal senescence and you interrupt it in the wrong cells, then you may lead to an abnormal lung itself. So lots of lessons to learn, and therefore we are still working on this, which I'm happy to continue to do. And what lessons did I learn along the way? Find a niche. You know, do something that others are not necessarily doing, because it's hard to compete against people who've been doing things for many, many years. Value your mentors. Be humble, as I explained. Build resilience. Pay it forwards. And sometimes if you have to, reinvent yourself. Find things to do that are different. And I'll end by saying, you know, you have to really value the people in your life. So I like this word, spuddle, to work ineffectively, to be extremely busy while achieving absolutely nothing. All right, many of you can relate to that, right? Some people work all the time, but nothing happens. And why is that? Because we waste time on things that are not helping us. So how do we change that? We have to stay focused. It's like poor Ulysses, who was tempted by the sirens. Stay focused. Stay focused, try to understand what you're doing, set rituals, set limits, and be able to continue to move forward. And you have to strike that balance between work, all the things that we do, and life, which is should be here, but it isn't. Life, yes, there's life. So life is very important. And I see life as a pie. You know, what is the flavor of your pie? So you can't just do work. You have to do other things, spirituality, advocacy, which I'm really focused on these days, family, including pets. I love my pets. Hobbies. You have to have hobbies, well-being, etc. So all the wonderful people travel, things like that. This is my wonderful husband and my children. My son's about to have a baby. I have grandchild with my first child, and then here are my doggies. And also take care, be mindful, reflect. Every morning, I spend a moment of reflection and express gratitude, do a gratitude affirmation. What are you grateful for today? If you start the day in a positive way, you end the day in a positive way, and you can have more groundedness when things don't go well. Think of who makes you happy, your family, the people around you, and what they bring to your life. And that's what I've been blessed with and thankful for. And I want to thank you for your time and for your attention this morning.